All right, in this video we want to discuss one of the important traits of disease, which is tissue damage. So, the first thing we should understand about tissue damage is really what defines disease. So, prior to disease, someone might experience an infection. And an infection is any time that a microbe enters the body and grows where it shouldn't. Right, so anytime a microbe gets into our body and grows, a microbe that shouldn't be there at all, that's an infection. Or if a microbe moves to a new spot in the body and grows, that's an infection. But basically the best way to describe an infection is any inappropriate growth of a microorganism. But infection is not disease. So again, what defines disease is this tissue damage. So disease is when an infection, that growth of those microorganisms, begins to damage host tissues or organs. And this leads to a deviation from health. So again, disease requires tissue damage. Tissue damage means there is disease. And we just want to think about how microorganisms cause tissue damage. So there's a few mechanisms by which microorganisms can cause tissue damage. We're going to talk about a couple forms of direct damage. In this video, we aren't going to discuss something called indirect damage. And indirect damage is really when our response as the human host leads to damage of our own tissues as we try to battle an infection. We'll talk much more about that in other videos. So again, we're going to really focus on direct damage. That is the microorganism harming the host tissues. We're going to look at a couple ways they can do that. First, we'll think about extracellular enzymes, or something we're just going to call exoenzymes. And then we'll also think about things called toxins. So we're going to look at two ways that microorganisms can cause direct damage to you, the host: exoenzymes and toxins. And again, we're going to save indirect damage for another video. But again, indirect damage is when your cell, your body harms itself in its attempt to battle an infection. All right, so let's start by thinking about these exoenzymes. So again, one way that a microorganism can cause tissue damage is through the use of exoenzymes. So an exoenzyme, what is that? An exoenzyme is just an enzyme. We know enzymes are proteins. And it's an enzyme that acts outside the bacterial cell. That's where the exo part comes from. So exo, outside, enzyme, protein that does a chemical reaction. So in general, these exoenzymes are going to break down or damage tissues. In general, the idea of these exoenzymes is that they allow the microorganism to get into new parts of the body. So by breaking down a tissue, they can penetrate farther into that tissue. We're going to think about a few examples over the next few slides, and those will be hyaluronidase, collagenase, coagulase, and kinase. So let's think about these exoenzymes one at a time, and we'll examine how they work. All right, so let's start with hyaluronidase. So hyaluronidase has something to do with hyaluronic acid, or hyaluronin, depending on where you read about it. Hyaluronic acid is basically a chemical that your cells use to hold cells together inside of tissues. Think of hyaluronic acid as glue that holds tissues together. The enzyme hyaluronidase is made, here's an image of some cells held together by this hyaluronin or hyaluronic acid in between the cells. Again, hyaluronidase is an enzyme, and this enzyme's job is to digest or break down hyaluronic acid. So again, this chemical is sort of gluing these cells together. If we look at another image, hyaluronidase, released by these bacteria, gets in here, digests that hyaluronin, weakening the connection between these two cells, and now the bacteria can penetrate farther into the tissue, expanding their habitat. So hyaluronidase is one example of an exoenzyme. The bacteria makes it, they secrete it. It acts outside of the bacterial cell on the host tissue, causing damage. Collagenase is a very similar enzyme. So collagenase is another enzyme. It's going to act on collagen. Collagen is another chemical in tissues that provides the tissue with some strength. Specifically, collagen's job is to provide sort of tensile strength so that tissues can't be torn apart. So collagenase is an enzyme that digests that collagen. If we weaken the tissue, now the bacteria can penetrate farther into that tissue. So here's another diagram where some vascular tissue is held together by collagen, right? Collagenase is released by the bacteria, weakening the connections. That disrupts the tissue, causing damage, and it allows the bacteria to penetrate through that tissue, right? So again, exoenzymes all fall into this category. They're proteins that have enzymatic activity, and they degrade some aspect of the human body. They cause a deviation from normal health. So the next exoenzyme we should think about is coagulase. So coagulase, again, ase means enzyme. This one has to do with coagulation in some way. So let's think about coagulation. When blood clots form, we call that coagulation. They form when this chemical fibrinogen is chemically converted into something called fibrin. And then the fibrin sticks together and causes a blood clot. So fibrinogen doesn't stick to other fibrinogen molecules, but fibrin will stick to other fibrin molecules, leading to coagulation. So this coagulase enzyme, it triggers coagulation by converting fibrinogen to fibrin. We actually can see that in the microbiology laboratory. So these two tubes both have blood plasma in it. So blood plasma is all the liquid associated with our blood that has had the red blood cells removed. Things like fibrinogen are still in there. So these two tubes had bacteria put into the plasma, and the one with a plus has bacteria that do produce coagulase. They've caused the plasma to coagulate together, whereas the negative tube has bacteria that do not contain the coagulase enzyme. And the plasma is still liquidy. It still spreads out in this tube when it's tipped on its side. So again, one enzyme that microbes can have to harm the host is coagulase. And you might go, well, forming blood clots is normal. How is that disease? If a blood clot is formed when your body is not trying to make a blood clot, well, that is harmful to the host. So that is part of disease. The tissue is not in its normal state. But the next question becomes, well, why would a bacteria want to form a blood clot? The last two examples of exoenzymes that we talked about allowed the bacteria to penetrate deeper into a tissue. How does forming a blood clot help the bacteria? And here's the way I describe it. If I'm a bacterial cell and I'm in the human body, one of my goals is not to be noticed or detected by your immune system and your phagocytes that might try to kill me. And so if I can form a blood clot over myself, I sort of hide from the immune cells in your body. So I think of cells that have coagulase. They have the ability to sort of build a camouflage blanket over themselves. And as a phagocyte is looking around your body, it will see a blood clot and go, well, blood clots are normal. Those happen from time to time. And it won't recognize that there might be some bacteria hiding in or under that clot. All right, so that's coagulase. Let's think about one more, and this one is called kinase. And kinase is a very generalized name for an enzyme in biology. In general, kinases are just enzymes that add phosphates to other proteins. And so kinases, when they add these phosphates, they tend to turn other proteins on or off by adding a phosphate. So in microbiology, there's really two more specific kinds of kinases that we talk about, and they are staphylokinase and streptokinase. So these are kinase enzymes that Staphylococcus and Streptococcus bacteria have. And their job is to activate something called plasminogen. So the kinase turns on plasminogen. And the question becomes, well, what does plasminogen do? Plasminogen digests fibrin clots. So bacteria that have these kinase enzymes are able to digest clots or break clots down. So this raises an important point we should recognize, and that is that not all bacteria have all of these exoenzymes. A bacteria that has coagulase might not have kinase. A bacteria that has a kinase might not have coagulase. But certainly some bacteria might have them both. And I think it's important to recognize that a bacteria would not use both of these enzymes at the same time. That would be a waste, right? To both build and break apart a clot at the same time. So bacteria have the ability to choose which tools they're going to use at any given time. And one of their tools is kinase. Kinase activates the process of tearing apart fibrin clots. All right. So those are the examples of exoenzymes. Enzymes, proteins that act outside of the bacterial cell. Let's next address toxins. So toxins are non-enzymatic chemicals, and they're chemicals that are poisonous to other organisms. So we call some things exoenzymes, and then there are these non-enzymatic things called toxins, just poisonous chemicals that microorganisms can make. And we'll talk about a couple examples, exotoxins and endotoxins. And so toxins, again, are just chemicals that the bacteria make. The exo or the endo sort of refers to where they're utilized by the bacteria. So let's start with an exotoxin. Exotoxins are made by the bacteria. They're actively secreted and they act outside or exo the bacterial cell, right? So we call it an exotoxin because it's made and sort of spit out. It acts outside the bacterial cell, just like the exoenzymes did. There's a whole bunch of examples of exotoxins, and hopefully we'll get the opportunity to talk about several of those. We won't address those right now. But let's end by thinking about endotoxin. So exotoxins are made, secreted, they act outside the cell. What's up with an endotoxin? So endotoxins are made by the bacteria, and they're held inside the bacteria. So that's where their endo comes. These chemicals are they're structurally part of the bacteria. So these chemicals are inside the bacteria. So let's think about an example on how this chemical can become toxic. So one example, and the most common example, is something called LPS, or lipopolysaccharide. We've talked about this in the past. LPS, this chemical over here, is a part of the gram-negative outer membrane. So we know that only gram-negative cells have LPS. It's structurally part of the outer membrane. And here we're saying that LPS is an endotoxin. So it's part of the cell, it's within the cell, but it's toxic. And what makes it toxic is that this chemical, LPS, causes fever when it's shed from cells. And the reason it would be shed from a cell is because the cell died via bacterial lysis. So when a cell dies and this chemical is spread all over the body, it can trigger a fever, and that can be damaging to the human host. We'll think more about that later when we talk about fever, but that's where we'll leave it right now. That's a good discussion of exoenzymes and then the toxins and how they harm the human host. So as always, I hope if you have any questions, you'll let me know, and I'll talk to you all again very soon.